In order to assure the biocompatibility of implants, we, biomedical, biomaterial scientists and engineers, must address not only aspects which promote normal wound healing and new functional tissue formation around the implants, but also we need to address how to avoid and or minimize outcomes which are undesirable because they endanger the welfare and sometimes even the lives of recipient patients. Such a crucial aspect is infection. We will start with a short review of what causes infection. Lecture number 20 is entitled A Short Introduction to Biofilm and addresses this specific aspect. We will start with a definition of what is a biofilm. A biofilm, sometimes colloquially referred to as slime because that's exactly what it looks like, is a community or a colony which is formed by pathogens, microorganisms such as bacteria and fungi, which multiply. These particular communities, the biofilm, contains sometimes the same or sometimes the different types of pathogenic microorganisms. These communities of cells of pathogenic microorganisms inside the biofilm are enclosing themselves by an extracellular matrix which they produce themselves. This extracellular matrix contains proteins, polysaccharides, and several other chemical compounds. The characteristics of a biofilm are that they attach themselves on solid substrate surfaces, and they could be either synthetic or natural materials, but a major requirement is that of having a moist environment. These pathogens, microorganisms, survive and grow even on tissues of living organisms. They are found even at their liquid interfaces. This is a very important distinction to make because the mammalian cells from tissues do not survive at their liquid interfaces. You may think of these microorganisms as pests which need to survive, and they have developed all kinds of clever ways which helps them in this particular search of survival throughout their lifespan. An example of a biofilm is that what the dentists call dental plaque. It is a slimy buildup of bacteria which of course live in the mouths, and this buildup is of course on the surface of teeth. This slide gives you some examples of bacteria which form biofilms. Of course, first and foremost are the Escherichia coli, some of which are benign and they're found in the gastrointestinal system of humans. Some of them can be pathogenic. Staphylococcus aureus is a microorganism which survives and is propagated in the air. Epidermidis, though, is a staphylococcus which is found on skin. Salmonella enterica, of course, is found in the gastrointestinal system, etc. This list, of course, is only giving you some examples. It does not exhaust the type of bacteria which form biofilms. To start with, a very important and almost frightening detail is that biofilm communities or colonies can develop within 3 hours from starting that particular development. There are several stages in the formation of biofilm. A summary of these steps is following. First and foremost, bacteria attach on surfaces in two steps. The first is an initial reversible form of attachment, but the second step is that of a permanent irreversible adhesion on substrate surfaces. Once the bacteria attach on a surface and they start proliferate, they form what we call microcolonies. Eventually, these particular colonies grow, deposit at extracellular matrix, which is a biofilm that covers this particular colony. This step is referred to as biofilm maturation. And last but not least, they have the capability on their own to detach from where they started the first colony, disperse, go somewhere else, another part of the implant, another part of the human body, and start forming a new colony and grow again. The schematic illustration of this particular slide shows the various steps in the formation of a biofilm. To start with, planktonic type of bacteria. These are identified as such because they have those little tails which are shown on the very extreme left side of this particular illustration. These tails, called flagellum or flagella in the plural, help the planktonic types of bacteria propel themselves, and as a result they can float or swim, if you wish, as single cells and approach a particular surface. At that particular point, within seconds, they make what I mentioned earlier is the reversible type of adsorption of this particular bacteria. As a result of that interaction, of course, the bacteria slow down and form these reversible associations with the surface. In the next step of this interaction, the bacteria adhere and form stable associations with the substrate surfaces. When they do that, they lose their flagella. At this stage, they start producing chemical compounds which are programmed by nature to produce, such as polysaccharides and other chemical compounds, and form an extracellular matrix. As shown in the middle cartoon of this particular slide, you see the cells now grow, and they form slowly but for sure three-dimensional structures. The growth brings about, this is within hours and days, brings about, or I should say traps, all kinds of other cells that may be floating around. They are not discriminating. They want to grow, and they grow in such a way, not only by proliferating themselves, but by capturing other cells and all kinds of other debris. And they form this particular colony on the surface of a substrate, which is the biofilm. This outcome is achieved within days and perhaps in terms of months from the initiation of this particular process. Another interesting aspect which I would like to bring to your attention is the fact that once the biofilm colony is formed, the cells which are inside that particular capsule, they become to specialize. And people have recognized and identified the so-called fixed cells, cells that are motile, therefore they have the capability to leave the colony and go further downstream and start a new colony. Some of the cells are referred to as pillars, some as monolayer cells, but usually the structure is multi-layered, and it is three-dimensional. This is the structure of a biofilm once it is formed on the surface of a substrate. I am including this slide for your information. It describes in words what I related to you in my discussion of the schematic illustration of the steps in the formation of a biofilm, which was presented in the previous slide. Because of their importance, biofilms have been studied extensively. There are universities in biomedical engineering, departments which have been specializing in studying of biofilms, not only for biomedical, but for other engineering type applications. In the following slides, I will summarize some of those important characteristics. First and foremost, there is no doubt that the biofilms are complex environments, and as far as the time course is concerned, they are very dynamic and they are changing with respect to time. The bacteria, which are the constituent cells in these biofilms, attach not only onto substrates but also to each other with the help of the extracellular matrix, which they produce and deposit on the surface of the substrate. The biofilm bacteria use chemical compounds which they are programmed to release, and that's how they communicate with each other. These particular colonies are very efficient in terms of cell-to-cell communication. The genetic expression by the bacteria in the biofilms is different than that of the planktonic. Remember, the planktonic forms are the ones that have the flagella. In addition, the enclosed environment which the biofilm provides maintains conditions which are related to nutrition, other biochemical compounds, which are required for the survival and growth of these bacteria communities. This particular slide illustrates some of these points. You see here the adhesion of the cells, and you notice that we have a difference and change from the planktonic into the adhering cells, the growth of the biofilm colony, and for the case illustrated on the extreme right, you see a huge colony, but inside this particular colony, you see many different types of cells. Remember what I mentioned earlier, that within the biofilm, the dwelling bacteria specialize, and they can become what we call fixed cells, pillars, monolayers, motile cells, etc. An important aspect which I would like to emphasize by repeating most of the previous word slide is the fact that the biofilm maintains the environmental conditions required for the survival and the growth of bacterial communities, even under bulk flow conditions. This particular situation is illustrated in this schematic, and you can see here an artistic rendition of biofilms, many of such colonies, which have grown on the surface of a substrate. You may notice that in the middle, sometimes these particular colonies become void of cells. This is a result of the limitations of transport of nutrients and oxygen that they need in order to survive across the biofilm and through the various layers of other bacteria cells. In this illustration, we have bulk fluid, which means flow of a particular liquid. It could be, for example, blood or some other body fluid. And you notice that in order to optimize the flow of the fluid, the structure of these colonies is such that creates not voids, I should say, open spaces through which the fluid, like blood, etc., can flow. Why is this aspect important? Don't forget, in order to survive, these growing cells need nutrients and need oxygen. And the flowing fluid brings this particular critical for the survival and growth nutrients and oxygen to the bacteria colonies in the biofilms. Another important characteristic of biofilms is that the bacteria in those colonies can lure other cells for food. In addition, and most importantly, from the perspective of their survival, as I said earlier, they have developed very clever ways to survive. When they encounter starvation conditions, either because those colonies have grown too big, or because something has happened at the site of the original formation of the colony, they take action. And the action is the following. They start forming planktonic cells. That means that those cells are motile and they can move. And they do move. Under those conditions, these biofilm communities start detaching clumps of the bacteria. They can move downstream to other parts of the infected organism, attach to other surfaces, and grow into new bacteria colonies or communities far away from the original site which started the biofilm formation. This slide is a schematic illustration which summarizes the life cycle of a biofilm, starting with the planktonic cells with the little flagella, which start attaching, make permanent adhesion on the surfaces, start forming the colonies, which under the biofilm protection grows and keeps growing, and finally, under certain conditions, they start forming planktonic cells, shown here on inside frame number 3, the little cells with the flagella, and then they become released and disperse themselves. They can flow downstream to another part of the implant or the body of the recipient host and start forming a new colony under the protection of the respective biofilm. Biofilms and their potential complications have been of importance not only to biomedical, biomaterials scientists and engineers, but also to our colleagues in the healthcare business, because they are associated with infection, and that particular aspect is a major complication that affects the welfare, the well-being, and sometimes even the endangers, I should say, the lives of patients. Some examples of biofilm-related diseases are shown here. Chronic lung diseases such as cystic fibrosis, ear infections, dental plaque, wounds, and in addition, complications related to implants, prosthetic bone implants, cardiac valves, catheters, etc. the reason that not only our colleagues in the medical healthcare field, but also we as biomedical engineers interested in implant materials and devices, the reason that we are interested is the following. The biofilm is a situation that protects the enclosed bacteria from any harm that might be instigated, let's say, by the immune system of the host. They are protected. The activated white blood cells cannot reach them. They cannot phagocytose them. They cannot kill them. In addition, they are protected from damage that may occur because they find themselves in an environment under flow conditions. Those dynamic conditions many times can bring about damage to cells and tissues and organs. They are not affected by medications which the recipient patient may take, such as antibiotics. Again, the antibiotics do not penetrate that protective biofilm under which now all these bacteria are found and are protected. Toxins cannot reach them. And if you think that as biomedical engineers we can take our favorite material and use bleach and detergents to clean and get rid of the growing bacteria, they are not killed by bleach and detergents. They have developed many interesting ways to survive in any environment in which they find themselves. This is the end of lecture 20 with the title A Short Introduction to Biofilms.